and we'll turn it over to Johannes. Johannes, go ahead.
Thanks, Meredith. Thanks, Charlie, also, and thanks for Rob, even though you can't be here, for inviting me to give a presentation today. So it's really a great pleasure to see so many of you, even though virtually, but joining this presentation. So yeah, I would like to talk today a bit about our research on ancient pathogens. I mean, as Meredith has just introduced, we're also working on human population history and on the evolution of humans. So comparing them, for example, with extinct hominins such as Neanderthals or Denisovans. Today, I really want to focus on that angle, the ancient pathogen work that we have started maybe about 10 years ago. And what motivated that research 10 years ago was that I realized that we know surprisingly little about the evolution, especially the early evolution of pathogens. So where did they come from? What was the zoonosis that first gave rise maybe to tuberculosis or plague? And how did they change through time? What's the mutation rate? What's the evolutionary rate? What's the host pathogen interaction? So how did they adapt to mammals or to humans? And how did humans maybe adapt to them? And the reason why we know so little about that, I also realized quite quickly, and that is, of course, we don't have fossils, right? Usually when we study evolution, for example, human evolution, we can look at Neanderthal fossils or Homo erectus or Australopithecines and kind of see at least how the morphology changed through time. And of course, we can do the same for many other species, like paleontology, for example, does. But for pathogens, this wasn't really done because nobody is using microscopes and looks for kind of pathogen particles from thousands of years ago to learn something about their evolution. And this is what we wanted to change with the molecular approach that we now call ancient pathogen genomics, where we basically go back in time, we take skeletons from the past and then reconstruct the pathogen DNA that might still be present in those ancient pathogens, and then reconstruct them genome-wide to learn something about not the shape maybe, but at least how the building blocks, how the blueprint has practically changed through time. How do we do that in the Mickey Mouse version? We take ancient skeletons, we extract DNA from bones or teeth. Teeth are particularly good because inside the tooth is actually dried blood. So if you have a blood-borne pathogen, for example, you might actually get it from the bone powder that you might drill out of an ancient tooth. That DNA is then usually extracted and prepared for sequencing libraries in a clean room facility to avoid contaminating the samples with our own DNA or other lab contaminants. And then that DNA usually gets sequenced in high throughput sequencing machines. And as you know, there was quite a revolution over the last couple of years with about 100 million-fold improve in the sequencing power of those machines only in the last 15 years or so, which really allows high throughput sequencing and then whole genome assemblies of those ancient pathogens as well. So what we gain then basically is some sort of molecular pathogen record. We basically are then able to directly look at mutation rates because we have calibration points from the past. They are radiocarbon dated, so we can say this is how it looked like 5,000 years ago, 1,000 years ago, 500 years ago, and then measure how it changed, this pathogen, through time, how many mutations happened for 100 years or even per 1,000 years. We can look how it interacted with humans. We can, for example, see, as I will show you today with Yersinia pestis, how it became adapted to mammals. So what kind of genetic changes happened through time that made it more efficient as a pathogen to infect mammals. And we can do that, of course, for other pathogens as well, and look at the human side. So the human side, I have to say, we are not as advanced yet as on the pathogen side. We are also starting to see how certain pathogens have actually shaped humans, as we know, for example, malaria, so Plasmodium has done, and many other pathogens, so that's yet a different angle. And of course, we can give some very important information for historians, for example, for archaeologists, like what caused large epidemics and pandemics, maybe even in the past. So which pathogen strain is actually responsible for, like the Black Death I will talk today about, or Justinianic plague, the plague of Athens, the Hittite plague, the famous Egyptian plague that even made it into the Bible. So what were those causative agents of those ancient pandemics? So this is the type of work that I mostly did with those three researchers. So Kirsty and Alexander were postdocs in my lab in Tübingen before I moved to the Max Planck Institute in Vienna. Verena was a PhD student of mine, and Kirsty and Alexander are now group leaders at the Max Planck Institute in Leipzig, where I now have my department. And Verena is actually a professor in Zurich right now, and is soon moving to Vienna. And we focused on different pathogens over time. So plague is really Yersinia pestis, the causative agent of plague, is one of the main topics. And I will talk mostly about that today as an example, what we can actually do in this type of approach. We also looked a lot into Mycobacterium leprae evolution, so the cause of agent of leprosy, where the oldest ones we could actually trace back now to even Egyptian mummies, so about 3,000-year-old samples. We looked into the causative agent of syphilis, Treponema pallidum, as well as Treponema pallidum patenoi that causes yaws, and we could look at that in the New World, in the Old World. It's a lot of hypotheses about when it emerged and was it brought by Columbus in 1494 to Naples, because that's kind of what the first historical records are, and we seem to not really confirm that idea. And if maybe people have some specific questions on that, we can talk about that in the discussion. We also looked a lot into Mycobacterium tuberculosis evolution. There was one really interesting question there, and that is, what did the tuberculosis look like that was before Columbus in the Americas? Because paleopathology showed that it was there. However, molecular biology showed that tuberculosis in the Americas today is only European tuberculosis. So how did it actually then come to the Americas? Did it maybe come with the Vikings, or was it some other kind of transmission? And what we could actually show was that it was indeed there before, but it was not the European version, but in fact it was a version that you today find in seals and sea lions. So it was actually a zoonotic emergence, probably already 2,000 years ago, that then disseminated within the human population and infected Native Americans. And then when Columbus came, European contact came, then it was basically replaced by the European version, which was probably more effective or infectious. We also looked at Salmonella enterica, so typhoid fever, which was, it seems, one of the first pathogens that was actually transmitted from the Old World into the New World. It was probably largely responsible for some of the large epidemics that happened among Native Americans. As you might know, and there are estimates that up to 95% of the Native American population was killed by germs, by pathogens that were introduced by after European contact in the first 100 years. And we were also quite lucky that we had access to the Iceman, so this frozen mummy that was found about 30 years ago in the Alps, and we had access to the stomach content. We could actually reconstruct Helicobacter pylori, which, as you know, many of you carry in your stomach, and that can cause ulcer or cancer. And we could reconstruct and actually show that the Helicobacter pylori that was found in the Iceman is very different than the one that you find usually in Europe today. It was more a version that today we find in Asia, especially in South Asia. Recently, we've also done quite some extensive work on virus evolution. So we could actually show that hepatitis B virus actually preserves quite well. It's probably one of the kind of best preserved pathogens in ancient human bones. We could even get it in bones that we usually don't get pathogens from, like a petrous bone, the inner ear, where the hepatitis B virus is also found, probably because people had chronic infections over many years of their life, maybe even getting infected after birth. And we just published a paper a couple of weeks ago that was led by Arthur Kocher and co-led with Denise Kühnert, a colleague of mine at the Yersinia Institute, and where we reconstructed more than 100 genomes from all over the world, some even from Native Americans that were up to 10,000 years old. We could really look at the evolution of this virus through time and see that there were some extinct lineages and replacements and lots of dynamics, and even some lineages that survived and only popped up quite recently in HIV patients, which is still kind of like something that we try to understand how that happened and where it was hiding for the last 4,000 years. But that could be a presentation of itself, and I don't really have time to go into detail here, but I just want to mention it. But today I really want to focus as an example of this ancient pathogen genomes approach on plague, on Yersinia pestis, the causative agent of plague, and what we have learned about this pathogen over the last 10 years or so. And the first thing I should say, it's actually not a good example of a human adapted disease, because it's not a human adapted disease, it's actually a rodent disease. It has emerged probably within rodents, and it's disseminated within many different rodent species. So there are many wild reservoir populations where plague can be found today, Yersinia pestis. And the way how it's actually transmitted is really interesting, because it's quite a nifty way how it actually jumps from host to host. And the jumping is quite literate, because the vector is actually a flea. So the bacteria have evolved in a way that if a flea bites an infected individual, it takes up the bacteria in its stomach. And inside the stomach, they actually build a biofilm that blocks the pre-stomach and the kind of proventricular, how that's called, kind of just entry of the stomach, which actually means that the flea cannot swallow more blood, and that actually causes the flea, of course, to get kind of crazy. It's starving, and it keeps on biting. And every time it bites, it sucks the blood and it comes into contact with the biofilm, but then it has to spit it out again because it can't swallow it. So then actually infecting with the other bite mark, then in the same individual multiple times, or then even another individual, and that individual has died. And such a starving flea bites up to 200 times a day, and it takes about 14 to 20 days before it starves to death. So you can imagine how kind of effective that is, because it kind of keeps on biting and infecting basically the bite mark, and thereby transmitting the bacteria. Eventually, the rodent might die, and then the flea is looking for the next warm body, and that warm body can be another rodent, but it can also be an animal that lives in close proximity with humans, such as domestic animals, or, for example, then commensal animals such as rats or mice that live in human habitation or close to humans. And then the same might happen there. If the mice dies, if the rat dies, if the dog dies, or the cat, or they come in close contact with humans, then the disease can also, the flea jumps to humans, bites the human, and then the human gets infected. And then the bacteria usually travel through the lymph system. And in the lymph node, they actually cause an infection. That's then kind of the bubo, this kind of swollen lymph node. And then through the lymph system, the bacteria can also then enter the blood, cause then sepsis. It's quite severe. And mortality is quite high. So if there's no antibiotic treatment, about 50 to 60% of the people that get infected die within 7 days to about 14 days or so. This is, however, not the only form, only manifestation, if you want, or the only entry of the pathogen. There's also different forms. So if people have bubonic plague and they have an infected lung, because sepsis then also, of course, causes kind of multiple organ failure, actually necrosis, which turns the hands and limbs black. This is why it used to be called the Black Death. But if then the lung also gets infected, and then somebody coughs up little particles of the lung and somebody else inhales them, and they can get something that is called pneumonic plague, where basically it's a transmission from human to human. That is actually a very severe form of plague. Often symptoms occur within 24 hours, and often people die after 48 hours to about 3 days. So that is actually so rapid and so fast that even antibiotic treatment is usually too late. So this is also why Yersinia pestis is researched in a lot of countries in the world by defense ministries, because it's regarded a bioweapon potentially, and of course it could be used by bioterrorists. So there's a lot of places in the world that research Yersinia pestis related to this potential use. And it was even, used in World War II by the Japanese when they bombarded China with blankets, with basically fleas that were infected with Yersinia pestis. And as you will see in a minute, it has even been used in the medieval time already as a bioweapon. So Yersinia pestis, or plague, is still around in the world today. So a lot of people think it's something from the past, but actually you find it almost in every continent. There's about 2,500 cases that occur within human populations, but it's very kind of rare. It's mostly found in rodent populations. Fortunately, today we have very powerful treatment with antibiotics, so mortality is really low, but untreated, of course, mortality would still be about 50%. And for pneumonic plague, as I mentioned before, it's even higher, so mortality is somewhere up to 90%. So it's really a kind of highly lethal pathogen if it would be, for example, inhaled. That could, again, be used in bioterrorism or something like that, sprayed in a subway, in a tube or something like that. That could be kind of potential misuse of it. Historically, people have reconstructed there were probably 3 pandemics in the last, say, 2,000 years or so that were caused by Yersinia pestis. The first pandemic was also called the Justinianic plague. It happened, or it started in the mid-6th century. It lasted for about 250 years or so before it disappeared in most parts of Europe. But then it came back in a quite fulminant, kind of large-scale pandemic that's called the Black Death. I will talk more about that in a minute. And then there was the third pandemic that started in the mid-19th century, actually in the Yunnan province, so close to Hong Kong in China. And that is also the time when the pathogen then spread all over the world, because Hong Kong, of course, was an international harbor, especially ship rats, which is Rattus rattus, so the so-called black rat. They are probably a good reservoir for the disease, and they spread it all over the world. So the reason why you find it today in Africa, Madagascar, in North America, South America, also in Oceania, is the Hong Kong plague, because then the ship rats really disseminated all over the world. The origin seems to be in Asia, but this is really when it then spread in all kind of different countries all over the world. Our first focus for our ancient pathogen work was the Black Death, because at the time when we started this research, it wasn't really clear what had caused the Black Death. There was much debate. Was it Yersinia pestis or a different pathogen, maybe a virus? Some people said filovirus or hemorrhagic fever. So there are all kinds of hypotheses. And that's something we wanted to test. The Black Death, as I said, probably the most famous, probably now after the corona pandemic in human history. It lasted about 5 years. It started in the mid-14th century. And it killed approximately half of all Europeans. So about 30 to 50 million people died within this 5-year period. Based on historical records, you can kind of reconstruct the entry route and the kind of way how it moved through Europe. It started somewhere in the east, in Eastern Europe. And the first historical records we have from Kaffa, which was at the time a colony by the Genoese. And actually this colony was under attack, but what they called the Mongols, which was the Golden Horde Empire. And during the attack, they actually launched dead bodies over the city walls. And it said that from those dead bodies, the disease was actually spreading, so potentially through then fleas. And this is why I said it was already used as a bioweapon in the 14th century then. And eventually the Genoese had to retreat because the plague was rampaging through the city. And on their retreat on ships, they actually took the disease to Constantinople, later on to Messina, to Genoa, and to Marseille. And then over the next year, it spread in the vicinity. And then you can actually see the different colors, the different waves that really spread all over Europe. It has been said that it likely originated in China. Currently, we think it's more Central Asia, but that was basically based on modern data. But now when we look at the ancient data, it seems to have probably emerged somewhere in the region of Kazakhstan today. As I said, it had been hypothesized that Yersinia pestis was the causative agent, but it wasn't really clear. And we really had to kind of test that directly by taking skeletons from the time and see if we can actually reconstruct the pathogen DNA. And indeed, we were quite lucky. We had access to a Black Death cemetery from London, the East Smithfield Cemetery. All the people there that are buried on that graveyard died during the Black Death, and we could use them to then extract DNA. But of course, it's not so simple by just taking a skeleton, extracting DNA and getting plague DNA. It's actually quite challenging because the vast majority of the DNA that you get from an ancient skeleton is not the pathogen, but it's the DNA from the environment, it's plants, it's bacteria. It's basically a soil sample because it has been in the soil for hundreds of years, and a tiny proportion is human, and even smaller proportion is of course the pathogen that killed the person than whatever 670 years ago. So we had to develop an approach that we can actually fish out the DNA that we're interested in. You could use that PCR, famous method that you could just amplify fragments, but of course amplifying a whole genome in tiny little fragments is really challenging. So what we used at the time was a DNA capture array, where we use hybridization capture. So we use the property of DNA to bind to each other if it's similar. And in this case, it was an array. Today we do it in solution, where you have the DNA on a little glass slide, which is the whole genome in this case of Yersinia pestis. And you then incubate that with the DNA from the ancient skeleton. Everything that is complementary will bind to the surface. Everything that is not, you can wash away, and you can then put that into a sequencing machine, high throughput sequencer, produce millions of DNA reads, and then basically assemble the genome in silico. And you can actually see how much Yersinia pestis DNA we had before enrichment, and after enrichment, you can see was quite powerful. So it was really quite successful, this type of enrichment. Again, today we don't do that in class slides, we do it in a solution, but it's still really efficient, and we can actually get enough DNA out of it. And this was then for this first samples that we have from the East Smithfield from London. We could get about 99% of the genome reconstructed, 30x coverage, which means we have seen every position of the genome around 30 times. What we can then do is phylogenetics. We could then kind of see how this Yersinia pestis genome that we have reconstructed from the Black Death is related to modern day Yersinia pestis. And this here is a phylogenetic tree of modern day Yersinia pestis, so the one that you find in the Grand Canyon or Madagascar or in Central Asia. Most of them fall on branch 1. This is really the most common type. Again, you find in the US or in Africa or in Madagascar today. And then there's other branches that are mostly found in Asia, Central Asia, China, Mongolia, and so forth. And the first question then, of course, was, where does the Black Death fall, right? And we really had no idea. Some people had hypothesized it would fall somewhere in branch 2. Some other people said branch 1. Some people said it's more like a deep divergent branch that we call branch 0. But where it actually falls, which makes a lot of sense now in the read-through perspective, is it falls on the base of the vast majority of strains that you find in the world today. It's falling very close to this multiforcation where a lot of genetic diversity was born. And of course, that makes sense if you think about the Black Death as a big pandemic like Corona today, where we have the Alpha, Beta, Delta, Omicron, or whatever kind of variant. And the same happened probably in the Black Death. So there was a lot of genetic diversity born. So therefore, the Black Death sits on the base of that. It's a bit like the Wuhan strain in a way, right? That gave rise to a lot of the genetic diversity of Corona today. What was interesting, though, is that it's literally the common ancestor. It doesn't have a single derived mutation. It doesn't have a single position that is unique to that genome. It doesn't have a single gene, doesn't even have a single position in the genome, which of course also tells us that every strain that is still present today that is born out of that diversity would probably cause the pandemic again, which basically means it's not more virulent, that genome, than the genomes that are still circulating in the world today, which is itself quite exciting and interesting. We then did a lot of follow-up work. This was mostly done by Matthias Spirou in my lab. So she collected a lot of samples from the post-Black Death era to see how they fit in the phylogeny. Maria was particularly interested in what happened after the Black Death. So the Black Death, 5-year big pandemic, half of Europeans die. But then there's about 7,000 plague outbreaks that happen in the next about 350, 400 years until the 18th century when plague disappears from Europe for good. Is that the same strain? Is it a derived strain? Is it a new introduction? Because a lot of people have hypothesized that plague was reintroduced to Europe multiple times. So every wave, is it then something that comes through the Silk Road or through some ships from somewhere in Central Asia, comes back to Europe, then gets replaced, and there's something else coming. But she wanted to test that. So she collected a lot of samples from a lot of places. And first of all, she got one sample, which was particularly interesting because it was from Eastern Europe, not too far away from Crimea. And this sample, surprisingly, actually falls one mutation basal to the Black Death. So it has one mutation less, but it's again the common ancestor. And that's pretty cool because the historical records say it came from Eastern Europe, and now the genomic data tells us the same story. So basically, we have it in the East, and then it collects a mutation, and then becomes the Black Death. She also had a number of genomes from France, from Germany, from Spain, from Great Britain. They're all from the Black Death, so from that 5-year initial pandemic. And they fall in the tree in this position. You can actually see they're all identical. They fall on spot with the London strain. So they're all identical. So unlike what we have with Corona today, where we have the Alpha, Beta, Delta, Omicron, during the pandemic itself, during the 5-year Black Death, the bacteria didn't have enough time to actually evolve. They didn't collect the mutation. They're all identical. So those 50 million people were killed by an identical bacterial clone, which I still find amazing that it happened. But now that we know the mutation rate of Yersinia pestis, it makes a lot of sense because we collect about one mutation every 10 years. So indeed, there wasn't basically enough time to collect new mutations. It might be that if we sequence more, maybe we find some variant, but so far everything, all the genomes we have, which is about a dozen or so from the Black Death, they're all 100% identical in their genome. Then she had a number of genomes which are from the 14th century, so just after the initial Black Death, and they're falling a bit on this branch here towards the third pandemic, so towards the strain that then basically disseminates from Hong Kong in the 19th century. And they're a bit like pearls on a string here. And this is interesting because we see this microevolution starting in Eastern Europe, then with the Black Death, and then in London, and then in Eastern Europe. So we basically see an evolution of that strain that later on gives rise to the third pandemic in Europe. So the third pandemic also originates from the Black Death, not from one of the other strains, which was also a big surprise. So that evolution basically happened in Western Eurasia, but then probably circulated back into Eastern Asia. She also has a number of genomes now that come from the post-Black Death period, so the 15th, 16th, 17th century, and they all come out of the Black Death, and they give rise to this branch here, which is the post-Black Death branch, we now call it, which is an extinct branch. It's not found in the world today anymore because it got extinct, because we don't have it anymore in Europe. It's basically eradicated in Europe. But it's all coming out of the Black Death, which basically tells us there was no back introduction. The Black Death was the initial event, and then it basically mutated, new variants arose, but they were all European. So there was a Western Eurasian, basically, then reservoir, which means there was some animal where plague was hanging out for a very long time. And then eventually we have the last strains just before it went extinct. Those have a very long branch, and it's actually interesting. If you look into their genome, we actually see that they have a deletion in common. Long deletion, about 50 base pairs are deleted. And in this region is actually some of the virulence genes. So those strains, the latest strains, they were probably less virulent than the earlier ones. And that's interesting, something, in fact, we also see for the Yersinia plague, where we also have some genomes, so some older genomes. So this happens at the end. Maybe that was one of the reasons, maybe the loss of virulence also caused somehow the loss of the pathogen within the human population, probably also the reservoir population. So in summary, what we can say here for the Black Death is we have this introduction in the 14th century, some microevolution in Europe, and then it circulates back into Asia, probably already starting in the 14th century. And then in the 19th century, it basically gave rise to the third pandemic strain, the one that had evolved in Europe. One question that we also had was what happened before those historical pandemics? So we have now genetic data from the Yersinia plague, from the Black Death, from the 19th century, but what happened before plague? Was that around or was it not around? When did it actually emerge? And that's a bit of a more difficult question, because if you have a pathogen that was circulating 4,000, 5,000 years ago, you don't know what to look for. You cannot use the fishing approach, the hybridization capture, because you don't know what to capture. So you have to use a more kind of approach that is more naive, like some sort of shotgun approach, where you basically look at everything at the same time and see all the pathogens that are potentially present there, which is challenging because the amount of genetic data that is produced by those sequencing machines now is so immense, it's so huge, it's billions of DNA sequences, that just a simple blasting of every DNA fragment would take years for a single sequencing. So this type of shotgun sequencer therefore needs a new approach. Unfortunately, we could work together with Alexander, who's a group leader with us now, and Daniel Husson, a professor at Tübingen, and they developed a new tool that's called MALT, which is a Megan alignment tool. So some of you might know Megan, which is a tool a lot of people use for metagenomics. And this MALT is a really fast aligner, and that basically allows a huge database, all the genomes that are in the world, to be compared with the sequencing, basically run of millions or even billions of fragments in a very short time. So about 1 billion reads in 24 hours. So it's super fast. It's about 500 times faster than BLAST. So therefore it doesn't take a year, but just a single day to actually analyze your data. And that was then extended by Felix and Ron, so a postdoc and PhD student in the lab, to then be actually able to use some of the properties that we have in ancient DNA. For example, ancient DNA is damaged, so changes through time, and we use that for authentication. And they then screened about 2,500 samples at the time and could indeed find, which was very exciting, plague preserved in prehistory. The oldest one that we now have is more than 5,000 years old, but we also have some that are 4,500, 4,000, 3,500 years old. Other labs also in Copenhagen found similar genomes, really old genomes from that time. So we could then do a phylogeny work that Ida in our lab, a PhD student, did. When you look at this prehistoric Stone Age plague, how we now call that, it actually branches off about 5,500 years ago from modern day plague. It has a really interesting and intriguing evolutionary pattern. And if any of you have an idea, maybe we can actually use the discussion for that, or you can contact me afterwards, why it evolves like that. I would really like to understand it, because what we see is that the oldest one evolves into the next oldest one, that evolves into the next one, and into the next one, into the next, and next, and next. So it's basically, it's like a backbone, an evolutionary backbone, where one pathogen then evolves into the next one, which is a couple of 100 years younger, which evolves into the next one, which is a couple of years younger. And it's really, it's one branch. And then basically they go and get extinct. And we have now more than 36 genomes from that time period, and they all behave like that. We basically only see one lineage through time, and it really seems to come out from one wild reservoir that is somewhere in the world and spills over into human populations. But then the kind of diversity kind of disappears, and then it evolves into a next strain, which means there must be a tiny reservoir. It really behaves like, for example, the flu, if some of you study that, So it's really weird, and we can't really explain it, especially not for a bacterial pathogen. But that's basically what we see, which is really exciting. But it has gotten extinct. It's not found in the world today anymore. And you can also then build some kind of like dissemination routes. So the oldest one was found here. That's the second oldest one, third, fourth, fifth, sixth, seventh, which kind of gives you this funny pattern how they might have moved through space and time, which in itself kind of looks strange if you would just take it like that. But it's interesting that we find the same pattern in the human DNA, how the human populations moved at the time. So it's almost identical. So the pattern how humans moved seems to be identical to the pattern how the pathogen moves. So probably humans moved the pathogen, either in themselves or in their domestic animals. And what's also really striking is that the Stone Age plague has a huge gap in its genome. And in the gap are some of the genes that are responsible or basically allow the pathogen to be transmitted by fleas. This particular strain would actually kill the flea. So it cannot live within the stomach of the flea. So it's not yet adapted to the flea vector. So this is something that could cause basically a pneumonic plague, but it cannot cause a bubonic plague. It could also cause sepsis, we think, because the close relatives can do it anyway, but it somehow has to enter, of course, the bloodstream, and pneumonic is probably the best explanation. Could also be gastrointestinal, but it's hard to test without actually reconstructing it, which is something we don't do, but we could theoretically do. So the other thing what we have, and that's really kind of the last thing I want to say, that is we also have now genomes from the early Bronze Age. They're about 3,500 to 3,900 years old, from Eastern Europe especially, and they actually, if you look in their genome, they have all the genes necessary. They don't have, basically they have an insertion probably, we think, that the other genomes, the Stone Age genomes, don't have. So basically something was inserted into the genome, which is the cassette that includes the genes that are necessary for the flea vector transmission. There's a whole bunch of other virulence genes that are necessary for the flea transmission that those genomes have from about 4,000 years ago, but the ones from 5,000 years ago, they don't have it yet. So it's really something that emerged on that branch here between the common ancestor with the Stone Age plague and then this genomes here. And we see quite some diversity popping up at the time. So probably there was even a kind of initial epidemic when this happened. So in summary, what did I tell you today about Yersinia pestis and how we used ancient pathogen genomics to understand the evolution of that particular pathogen? It caused 3 historical pandemics. That's maybe not new, but we now have really kind of confirmed molecular evidence for that. The Black Death genome has only very, very small differences to the modern strains. So it's not just that it's kind of the common ancestor, but modern strains only have about 50 to about 90 substitutions. So it's almost identical. So it didn't really change through time. That's actually true for the whole kind of family tree. They're all very, very similar with very few mutations. So it's all very closely related because it has a common ancestor only 5,000 years ago. The flea transmission itself evolved recently, probably between 4,000 and 5,000 years ago. There were epidemic outbreaks already in prehistory, and this becomes now really exciting because we can basically screen large collections from historical context to see if plague played an important role for large events. The Hittite plague I mentioned, the plague of Athens, Pericles died, Athens lost the war against Sparta, so maybe plague played a role. So there's all kind of questions we can ask now. We have probably an influence to this early migrations. So it's interesting that the moment that plague pops up, you see big dynamics, actually big shifts within the human DNA. So it could be that those pandemics actually change human migration patterns or influence them. And again, the earliest evidence for bubonic plague, so the plague that we find in the world today, is already 4,000 years old. So it's pretty old already. By that, I would like to thank all the people that did the work in the lab. So Kirsty, Alexander, the 2 group leaders, Maria, postdoc and PhD student, Marcel, Aida, and Michal, that were PhD students on those projects, the rest of my department. You can read more about that in the book which is published in the US that I wrote with a friend of mine. It's also available in many other languages if you prefer that. And otherwise, thank you for your attention. And I can see there's a lot of questions already in the chat. So.
Yeah, thank you so much. That was wonderful. We do have a lot of questions already in the chat, but feel free, people, to raise your hands virtually. We love to hear from you. And let me make it to where people can unmute themselves again. So we'll go first to Joachim Kurtz. You can go ahead with your question.
Yeah, thank you very much. A very fascinating talk. I was wondering how closely related Yersinia pestis is to Yersinia pseudotuberculosis. And in pseudotuberculosis, there is an effect of temperature on biofilm formation. Can you find out whether that happened somewhere in the history of Yersinia pestis?
So first of all, it's very closely related to pseudotuberculosis. It's in fact paraphyletic. So it's closely related to certain strains of pseudotuberculosis. So it emerged within the diversity of pseudotuberculosis. So it's one of the branches of pseudotuberculosis, basically Yersinia pestis. The divergence time, I mean, there's some estimates, the mutation rate is not completely constant. The molecular clock doesn't kind of work perfectly, but it's somewhere between 20 and 30,000 years that they diverged. I'm not sure if we should call the Stone Age plague Yersinia pestis or Yersinia pseudotuberculosis, because it does have certain elements of pseudotuberculosis. It does have, though, two plasmids, the Stone Age plague, that basically the pseudotuberculosis does not have. So therefore, we should probably call it Yersinia pestis 5,000 years ago, 6,000 years ago. There's a bit more diversity at that time. But again, what that kind of phenotype is, how it gets transmitted, that Stone Age plague, we do not know. About the biofilm formation that is temperature dependent, I actually don't know. I can't really tell you. I would have to read up about that. I'm not sure if the molecular mechanism is known, if the genes are known, and then we could basically test if those genes are present or absent, or if they have been deleted or not. But otherwise, I can't really say that.
Yeah, there's quite a lot known in Yersinia pseudotuberculosis about this process. Petra Dersch studies that, for example, and it would be interesting to look for those genes, I think, in the Stone Age plague. Yeah,
I mean, somebody could certainly contact me and we could look at those genes and look like if they're deleted, if they're there, what versions they have, if they have pseudogenized or something like that. That could indeed be interesting. I agree.
Great, thanks so much. Next, we'll go to Anne Ladissoet for a question. Anne, you can unmute yourself and go ahead. Oh, and it looks like you've gone back on mute. Try hitting mute one more time.
And how is this now?
Yeah. Perfect, thank you.
Okay, I was wondering what was your opinion about pestis minor, or the appearance or emergence of less virulent strain, or strains that are coming back after 20, 30 years of quiescence, and what would that be? What would that mean in terms of evolution? I say so because I investigated a plague outbreak in the DRC in February, and it was very mild symptoms, but everything pointed towards plague, with rodent die-off and 300 cases, suspect cases, but we have no confirmatory diagnostic. And it was really surprising not to see very acute symptoms. So yeah, I was wondering what was your opinion on that?
Yeah, of course, from the kind of archaeogenetic point of view, it's really hard to say anything about that, because we just have those fulminant kind of pandemic strains from the past where we have historical records and we have those large events. For modern day diversity, I'm really not a good expert to say anything about the molecular mechanisms that might cause the loss of virulence. I mean, we do have that loss of virulence in the past. Those things that we see in the past, we don't, however, see in the modern day diversity, which in itself is interesting, because it basically means that those strains with loss of virulence have gotten extinct. But it's something that one should investigate. I mean, I know my colleagues have been investigating some of the strains that were responsible for the larger outbreaks within humans, which was in Madagascar in 2017. And they have not really found evidence for loss of virulence in those strains. I don't know for the ones that you just mentioned that might be in Central Africa, and whether those strains are, for example, different. But it's something one could investigate. I mean, if you have some samples and we have collaboration partners, it would certainly be interesting to get more genetic diversity uncovered and analyzed. I mean, there could be a presence or absence of virulence genes. There could also be presence of absence of plasmids, for example, or genes from plasmids. And that all has been observed in various places in the world, so it's certainly possible, but for that particular case, I can't really say.
Yeah, because it's the Ant1 strain, so it's interesting. Yeah, we can discuss further.
Yeah, thank you.
I wanted to follow up actually and look earlier. I'm so intrigued by the flea transmission and the insertion you talked about. And I was wondering if you've looked at the virulence genes or anything that might give some clues about virulence associated around that time. Because if I understand correctly, one of the ways that fleas are really transmitting this zoonotically is when the host dies and they're jumping off, to a new host. And so you might expect an increase in virulence that would be associated with the flea transmission.
Yes,
And
so
increase in virulence in the natural host.
Yeah, no, exactly. I mean, that's exactly what we see in a way. We see, starting around 4,500 years ago, that those virulence genes that are necessary for flea transmission all together, it's actually a whole cassette of them. There's maybe like 12 known, and basically 6 of them pop up within a very short time. And by 4,000 years, we have them all in present.
I see.
So
those are on that same cassette with the.
It's not. I say cassette, but it's actually not what I mean. It's like there's a cassette indeed on the PMT, one of the plasmids, but the other things happen in other parts of the genome. And some are already in place. It's basically not that all the genes happen in one area of the genome. Which in itself is interesting that it's such a rapid evolution, but it's probably a whole mechanism that has to evolve before it can really kind of flesh out. And it seems to really, and this is mostly kind of data that we're just generating, that at that time you get quite some diversification. So it really seems to kind of spread within a very short time, around say 3,500 to 4,000 years ago. And then this loss of virulence we really just see at the end of the pandemics, and we're not quite sure why that actually would be. What is interesting, and I didn't talk about that, but the region that gets lost, those 50,000 base pairs, that is actually getting lost independently in the Justinianic plague, so the first pandemic, and the second pandemic, the Black Death at the end. It's also the genes that are coding for flagellum. So it's like the leftover genes from the flagellum apparatus, which in itself is again interesting because Yersinia tuberculosis, we just talked about, the closest relative, has a flagellum, and Yersinia pestis does not have it. And of course, flagellum is something that the complement system, the human immune system, the innate immune system readily finds and attacks. And that's of course true for other mammals as well. So it could be that loss of virulence is actually not a loss of virulence, but it's just an adaptation again to the host. So there might be in the region also virulence genes, but maybe it's also just an adaptation, so it doesn't get recognized by the immune system as much, because sometimes, of course, even pseudogenes or genes that are not as active anymore might be produced and might then be recognized by the immune system or something like that. So there's a bit of a debate if it's actually a loss of virulence or maybe it's just an adaptation.
Great, thank you. Next question we have is from Fred Fox. Fred, you can unmute yourself and go ahead with your question.
Okay, I'm in a rural area with a bad internet connection. So are you able to hear me the audio?
Yes,
yep, sounds good.
Okay, so I'm in New Mexico, and that's where most U.S. cases of plague occur. And apparently, from what I understand, the branch 1 entered San Francisco in late about 1899. But yet now it involves a number of vectors, a number of flea species, and a number of hosts, including lagomorphs and ecomongolates. It would imply to me it's a really good generalist, or there's been enough apt change for it to cross over to so many vectors and alternative hosts. And I worry about what that implies in terms of possible episodic periods when a crossing over to other ungulates, specifically domestic stock.
Yes, no,
have enough sequencing in the Southwest been done to indicate this change?
Yes, and I mean, I also saw your question here, so I think there were some problems in the connection, but I think I got your question quite well. So no, I would say there isn't enough sequencing done. There's never enough sequencing in a way, says the geneticist. But I think indeed there's not a lot of genomes, unfortunately, published so far, because often it's just the type, then it's branch 1, and then it's not exciting because it's a really deep branch 0 strain, and it gets sequenced genome-wide. But there's actually not a lot of genomes that has been sequenced from that region. It's mostly branch 1 indeed, and that evolution or that kind of diversity is kind of pretty well understood. It has entered indeed in the late 19th, early 20th century. It is part of this Hong Kong strain that basically that was disseminated during the third pandemic. Yersinia pestis itself has been found in almost all domestic animals. So it can jump to cats, dogs, ungulates. It's actually kind of it doesn't really have that barrier. It's kind of, it can indeed infect different fleas. But what is kind of special in a way is that there are some rodent populations that tolerate it in kind of some way, and that can serve as a reservoir, because in a way, when it, for example, disseminates within a rodent population like rats or mice that are closely living with humans, it's actually causing high mortality in those rodents, which mainly means it would burn out the pathogen. So therefore, what you need is a reservoir. You need actually an animal population where it can reside for a very long time and then re-emerge. Because of course you have a certain amount of immunity after infection. You know, half of the people die, half of the people survive. The people that survive probably are immune for quite some time, like we have currently with Corona again. And so therefore it can kind of periodically in these waves kind of come back. But it has to basically reside within a reservoir population. And those reservoirs have to form and it has to adapt. And indeed, it's interesting that prairie dogs, for example, today are a reservoir population in the United States, where it resides today, but there needed to be some sort of adaptation. It's actually something, again, I was actually curious about talking with my colleagues in Madagascar, where the black rat has become a reservoir, which it has not been in other places. At least people have speculated about that, because the mortality in rats is really high. But also that work has been mostly done on brown rats, not on black rats. So in fact, it could also be that the black rat naturally has been a good reservoir. But you need to find, basically, the pathogen needs to find a reservoir population where it can reside for a very long time. So therefore, it is indeed kind of easily kind of transmitted into other fleas and mammals, but it needs to find a reservoir so that it can actually reside for longer time periods.
Great, thanks so much. So going to your question about kind of that backbone shape of evolution of the Stone Age Yersinia, Mark Iravani in the chat asked whether you thought that could be a reflection of multiple bottleneck events that the human populations have gone through. And Cynthia Bell thinks that hypothesis makes sense. So interested to get your thoughts.
Yes, indeed. That would be, one explanation, but we don't see bottlenecks occurring in such a massive way, because it would really be, basically the genetic diversity has to be reduced to a tiny population that the backbone only survives. It's a possibility, but again, the human population is not a good reservoir. It would have to be an animal that is in close contact with humans. So it could be a domestic animal, but for domestic stock population, we don't really have good evidence for shrinkage or multiple events at that time period, because domestic herds are actually in the millions already in the Bronze Age. So I would really bet that it's some sort of wild reservoir. It's a rodent, it's a bird, it's some other animal that we don't have on the radar. And actually a bird would fit well, because birds could disseminate it over large distances. And then kind of they all go back, say migratory birds, they actually go back to this one lake in whatever, Tibet, or in somewhere in Anatolia or the Middle East, or even Africa or so, where the reservoir population resides, and then they basically bring it back, because that's exactly how the flu works, right? The flu usually in Europe or in Eurasia comes from East Asia and then moves over kind of migratory birds into the north during the summer, and then basically in the winter then kind of flies back to a lot of other places, and then can actually recombine, of course, with human and pig, for example, versions. But that's usually how it kind of moves through Eurasia. And something like that I could imagine as a mechanism. Yersinia pseudotuberculosis, the closest relative, is actually found within birds. Yersinia pestis has not been found in birds. But again, it's the Stone Age version, so it's a very different pathogen that's, as far as we know, extinct today. So maybe it was happening like that. But it's a 3,000 years time period where this backbone exists. And we have no reason to assume that it's an adaptation that actually happens, where like today with Corona, where you have a better virus, the Delta is better than the Alpha, so therefore it replaces all the Alpha. But here, I think that's not happening. It really seems to be indeed bottlenecks, but not bottlenecks within humans. And I think the bottlenecks occur either with this tiny reservoir that humans come in contact with, or it's basically episodes, but it has to be some animal population. But what it is, we don't know. But we would really like to know, because then we would understand this Stone Age plague, because currently there's just lots of wild speculation. We really don't know what it is.
Great, thanks so much. So we have time for probably one more question after the one I'm about to ask. So if you have a question, raise your hand now to join the queue. I'll go to one from the chat from Samantha Kistart. This is a bit of a different question, but she was wondering about the shotgun technique that you're doing and whether that also captured commensal organisms, and whether anyone has been investigating whether there's any indication that the commensals have also changed, possibly due to interactions with ancient pathogens.
Yes, absolutely. So microbiome work is done especially by Tina Warner in my department. She's also a professor in Harvard. You should invite her to give a presentation. She's one of the best speakers I know in the world. Like, truly, she's really amazing, really impressive. And she's doing microbiome work through time. And she basically looks at, for example, dental calculus, which is a biofilm that's, of course, forming on your teeth, and looked at Neanderthals and apes and compared them through human, basically, evolution and history. She also looked at coprolites, so feces from kind of past population, and looked how those changed through time. So this is something that's kind of an active field of research, microbiome, basically signatures through time, and it's really exciting. I can talk about that myself, but you should really invite her and have her talk to you about that. It's really exciting research.
Wonderful, thank you. All right, well, I think I'm going to turn it back over to Charlie to close us out.
Okay, thank you for a great talk, Johannes. That was fantastic. Really appreciate your time presenting to us, sharing your results with this group. And thank you very much, Meredith, for organizing. Great job. Before we totally break off to our next Zoom meetings or whatever, I want to just quickly share with you our last upcoming talk. Hopefully you're seeing my screen now in our Club EvMed webpage. We just had this great talk that we heard from Johannes. And then coming up on December 14th, we'll be hearing from Aida Andre on genetic adaptation to zoonotic viruses. How did chimpanzees adapt to SIV, HIV infections? Looks to be another great presentation in our Club EvMed lineup. And then after that, we have, we're working on the schedule right now for winter and spring. And so please stay tuned. We'll have talks about every 2 weeks or so. Okay, thanks again, everybody, for joining. Great to see so many people. And we hope to see you on the 14th for the last talk of this series this semester. Thanks.